Okay, so you have used those methods, no? Cytometric method, no? Using selective media, using the pipette method. But then how do you identify? Okay, how do the scientists know which plant to take? Okay, so let's look at them. So you can either use morphologically, okay? If you look at me, I'm half looking. I have the looks of my father also and my mother also. Okay, something like that. So morphologically, they will have a look that is in between the parents. Okay, there, see there, it's written intermediate between parents. Okay, so for this purpose, the vegetative and floral characters are considered. They include the leaf shape, leaf area, morphology, shape, etc., size, structure of the seeds and all. Okay. So for example, you have to take many features. Okay. My face is like my dad, but my hands, they may be like my mother's. Okay. So you cannot just look at the face, you cannot just look at the flower and say, oh, it's a belong to this species. No, it's in between this. You have to see all, you have to see many. So you can use isoenzyme analysis method. Isoenzymes, these are enzymes. It is an enzyme which can catalyze the same reaction of another enzyme. Okay, isoenzyme is like suppose this is enzyme 1 and this is enzyme 2, enzyme 3. Okay, enzyme 1 can do a function which will produce this protein. Okay, but whether enzyme 1 or 2 or 3 acts on it, the same protein will be produced. You get it? Okay. So hybrids, they contain isoenzymes of both the parents, okay, of one or both the parents simultaneously. Okay, so there are some unique isoenzyme. Okay, let's say from species 1 it has a unique isoenzyme, all right, and species 2 has no unique isoenzyme. So if you get the hybrid and you select with the enzyme reaction, then you saw that the hybrid could produce that particular protein, so you will understand that it has taken up the unique isoenzyme of one parent, okay, into the new hybrid. Okay, chromosome constitution. So you can just directly count the number of chromosomes, because from the ploidy itself, no, if it's 2n and you get 4n, you know that, oh, this must be a hybrid. Okay, okay, then you can do molecular techniques. Molecular techniques, it's when you how do I say, you okay, you have extracted a DNA, and then you have extracted a DNA, then you have isolated DNA from a particular plant. Okay, you've mixed and you added all the enzymes and all, no, then you place it in the gels. Okay, then you run it in the electrophoresis. Okay, an electrode. Okay, which then this the genes, they will travel. Okay, then because as this gel gets thicker down, it gets thicker. Okay, so the heavier ones, they get stuck, the lighter ones, they can travel really, really far. Okay, so this is how you can differentiate between the genes. Okay, you can differentiate between the genes of different plants, because each molecular, what to say, each this one, RFLP, random what is this? This one, no, RFLP is restriction fragment length polymorphism You can go through it in your own time if you want to go through, but you will learn more about this in the masters. Okay, so each RFLP of every species is unique. Okay, because nowadays, no, we mostly, the scientists, they use molecular methods only. This one has this pattern, this one has another pattern, this one has another pattern. Okay, species A has a very unique pattern, B has a unique pattern, C has a unique pattern, species D has a unique pattern. Okay, now we don't use microscope method anymore. We use molecular methods. Okay, it's the most dependable. Okay, so you can don't Google. I think you can YouTube. If you really want to learn more about this, you can YouTube. Okay, then even the chromosome number, no, in the hybrids will be more than the parents. Okay, but there can be problems, no. Sometimes 3 or more protoplasts can fuse. Okay, because that chemical doesn't know, hey, you come and fuse with you. Sometimes instead of A and B fusing, A and A fuse. Remember that heterokaryon, homokaryon, B and B fuse. You get it? So there can be some problems. Okay, by the chromosome number. Yes, yes, the chromosome number is 4n, here also 4n. But what is the point? No, this is AA and this is BB. We want AB, okay, 4n. So it doesn't mean that just because it has doubled, chromosome number has doubled, doesn't mean that it is the correct one. Okay. So these are some examples of plants which have been protoplasts have been fused. Okay, and their chromosome number. Okay, so you can just take 1 or 2, whichever it is that you can remember. This one is tomato, I think, 72. This is tobacco, 96. Tobacco, the 2 different species. Okay, so you can remember whichever is easiest for you to remember. 1 or 2 example is enough. Okay, so what are symmetric and asymmetric hybrids? If the chromosome number in the hybrid is a sum of chromosome of 2 parental protoplasts, the hybrid is said to be symmetric. Okay, so if 2n plus 2n you get 4n, means the sum of parents is equal to the chromosome number of the hybrid, so that is symmetric. Okay. Asymmetric ones, they will have abnormal or variations in the chromosome number. Okay. So instead of 2n plus 2n, you are getting 3n somewhere, you are getting 1n somewhere, then suppose instead of 2n and 2n, another 2n, you are getting 6n. So these are asymmetric ones. Okay. So just like in the table, you saw that there was a range. Okay. Let's see. Here in the table, this one, you're getting some range, no? So you add them up. Okay, if they are coming together, means they're symmetric. If the numbers are coming different variable, it's asymmetric. Okay, so what are hybrids? Cytoplasmic hybrids without any nucleus. Okay, so this is A and B. Okay, then you add some fusogen, then you fuse, but then nucleus somehow they escape. Only the cytoplasm of A and B are fused. So that is your hybrid. Okay. Okay, so hybrid and somatic incompatibility. Okay. So when you cannot fuse the protoplast of distantly related plant species, okay, because the instability of the dissimilar genome in a common cytoplasm, it cannot survive. Okay, suppose you want to cross a dog and a elephant. They are so distantly related, no? Or a parrot and a tortoise. One is a bird and one is a reptile. Okay, so they are so distant related. Okay, but obviously don't give those examples. You have to give plant examples. Okay, so when you try to combine and try to fuse a species of a plant which is let's say orchidaceae and an algae, okay, or a bryophyta, they are so distantly related. It's so difficult, no? So even if you are able to fuse them, it will unfuse itself, or it will break down, no? So this is referred to as somatic incompatibility. Okay, so you cannot produce hybrids when they are so far apart. Okay, or even if you do produce them, they will not be full-fledged. Well, they may survive maybe up to a certain stage, but they will never grow and reproduce. Okay, so they may suppose you are finally able, okay, a very distantly related species, you can finally make a hybrid, but they may show some abnormalities. Okay, they may show some abnormalities. Okay, so you need to really interbreed and crossbreed, and the generation of generations you may have to grow them so that you remove all the abnormalities. Okay, let's look at genetic recombination in asexual or sterile plants. So there are certain plants who cannot reproduce sexually. Okay, just like orchids, they prefer to reproduce vegetatively. Okay, because their structures, you have seen, no, to pollinate also is very difficult to open the flowers. Okay, and even if they do somehow pollinate, the embryo never survive. All right, so if you want to recombine, if you want to combine the genes, okay, one male plant A and female plant of B, so somatic hybridization is a good approach. Okay, when when you want to create a combined morphology or genotype of asexual plants. Okay, so how to overcome incompatibility in the sexual incompatibility? So it will help you to overcome the incompatibility. All right, so this is one of the uses of somatic hybridization. So some more application is you can transfer genes. Okay, so I think this is under applications. So there are some more applications. You can get disease resistant plant, environmental tolerant ones, you know, when you combine let's say this one has disease tolerance, then environmental resistant. Okay, this plant doesn't have. Okay, but this is an important food crop. Then you just fuse, and then you're getting a food crop also, and which is disease and environmental resistant. So there are some more applications. You go through by yourself. You know, children in mono. Then I've given some limitations also in the note. So thank you. Completed.